Hi ladies, this is chapter 17 on viruses. So these are the big important points from this chapter that you need to understand. So number one, what is a virus? So a virus is an infectious particle consisting of little more than genes packaged in a protein coat. So if you look down here, you can see that each of these viruses has genes, which could either be DNA or RNA. Okay, and it has this protective protein coat on the outside. In addition, sometimes they even have this membrane envelope around them, which I'll talk about in a little bit. The protein shell is sometimes referred to as a capsid. So one of the important things to understand about viruses is that they're obligate intracellular parasites. So they need a host in order to replicate. They can't do it by themselves. They need to get into a host cell in order to make additional copies of themselves. So and each virus has a host range. So every virus can only infect a certain number of cells that are specific to that virus. It usually has to do with how these little receptors on the outside. So which cells can the virus latch onto? For example, bacteriophages, sometimes referred to as phages, are viruses that infect bacteria. So they're not going to infect other cells. They're just going to infect bacterial cells because they have the parts in order to get into a bacterial cell. So I'm going to go through the two different possible cycles that a virus can go through. So the lytic cycle, which is consists of attachment. So the virus is going to land. We're going to focus on bacteriophages for this example. This goes really, really well when you're looking at the Hershey and Chase blender experiment. So this goes hand in hand with those notes. So here's the attachment. Then the virus is going to inject its DNA into the host. Other viruses may inject RNA, depending on the type of virus. Then it will commandeer the machinery of the cell, in this case a bacterial cell, to make copies of that virus. So synthesis of viral genomes and protein. They'll be assembled together, making sure they look exactly like the parent. Okay, so that's what those genes in the original virus would code for, how to make a new virus. And then finally, lysis will take place. So those viruses will be released from the cell. The lysogenic cycle is a little bit different. It does require, it requires a bacterial cell, but it's more of a dormant phase. So the bacterial genome will incorporate itself into the bacterial, or the virus's genome will be incorporated into the bacterial genome, and it will just kind of hang out. Usually it waits until the organism is under stress or a certain number of copies have been made. We usually refer to this DNA as a prophage. Okay, and then when that organism has made enough copies or is under stress or whatever, the DNA will then be used to make virus copies, which will eventually cause the cell to lyse and release the phage out into the world to infect more cells. Viruses have evolved over time, and I'm just going to go through a couple of really important things that you need to know for the AP exam. So number one, viruses, many of them now have viral envelopes. So this is derived from the plasma membrane of a host cell, and it has glycoproteins on the outside. And some of them are now using different genetic materials. So the viruses could have DNA, they could have RNA. Viruses that have RNA, such as HIV that causes AIDS, actually use retroviruses. So, or they're called what we call retroviruses, and they have RNA, and they use reverse transcriptase to actually turn that RNA into DNA and then use that DNA in the cell. So let me show you how that works. So this is how HIV gets into the cell. And you can see that it has this viral envelope around the outside with glycoproteins. These glycoproteins are usually derived from the host cell that it lives on. And so the viral envelope will fuse with the host cell's membrane, pushing in the genetic material. This genetic material then used in the case of HIV reverse transcriptase to turn to turn RNA into DNA. So you can see here's an RNA DNA hybrid, and then finally DNA is created. This DNA can then be incorporated into the nucleus. The virus will typically then wait for when the host is under a lot of stress. So usually whenever the individual gets sick, something like that, and that provirus can then be used to construct mRNA. Then mRNA can be used to make proteins. Okay, so the proteins will then be used to construct a new virus. Okay, that RNA will be used as the genetic material to put inside that capsid, and then it will be coated in a new viral envelope and released. So please let me know if you have any questions on this information, but this goes really well with the Hershey-Chase blender experiment, and I think that's all on viruses. Have a nice day, girls.